morning everybody. You see, in today's class, we will be dealing with biomolecules. So basically, what are biomolecules? Molecules that perform various functions within a cell. Okay. So these molecules have been divided into organic and inorganic. And you know, the total molecules present within the cell are known as cellular pool. Now you see, this organic and inorganic substances are there. Organic includes carbohydrate, fats, proteins, and the inorganic part includes minerals. These are ions and salt. Okay, minerals generally these are present, inorganic one in the form of ions, and salt is also there, and another major component of the inorganic compound is water. Okay, this water forms the main bulk. Now it has been found out or isolated that 70 inorganic substances are present in plant and 24 inorganic substances have been present in the animal. Okay, now you see what is the method? How do we study these biomolecules as a whole? How do we classify them as organic and inorganic? To do that, see, we make use of living cells. Any particular cell that is crushed. You know, it has crushed by a pestle and mortar. You see here we use a pestle and mortar and it is grounded. It is rather grounded and we use a trichloroacetic acid. Okay, so trichloroacetic acid is used and it is ground with the cell. So we get a slurry. What is slurry? Slurry, it is a thick liquid. Okay, that thick liquid is known as a trichloroacetic acid and that is filtered. Okay, that is filtered. You see, we take a test tube, we plug it with cotton, we have a funnel out here, we use a filter paper and we pour this slurry liquid. Now, after pouring that, we find that we have a filtrate here and we have a residue over here on the filter paper. So we consider it as acid soluble pool and this is known as non-acid soluble pool. Now this non-acid soluble pool here we categorize them according to their molecular weight. Now how do we do that? See, this filter contains molecules. See, acid soluble pool contains molecules. Those are approximate 18 to 800 Dalton by weight. Okay, 18 to 800 Daltons by weight. So that is considered to be as a micromolecules. Now they consider, what do they have? These micromolecules, they have monosaccharide, amino acids, certain other DNA strains, RNA strains like that. Now what is up here? That is known as non-soluble acid pool. They are very heavy and they are categorized as micromolecules. Okay, micromolecules and their weight is 10,000 or more. So we have 4 major organic substances here that is isolated, that is DNA and RNA, lipid, polysaccharides. Okay, these we have 4 things. And out of that, one thing we find that lipid, if we weigh this lipid, it is 800 Dalton. So that means if it is 800 Dalton, it would be here in the acid soluble pool, but it is not there. It is found in a non-soluble. Now how does it happen? You see, when this living cell is already pounded with pestle and mortar, we find that lipid is generally found in the cell membrane or inner organelle structure. So when they are grounded together with this acid and water, it converts into vesicles. So these vesicles are insoluble in water. Now you see this trichloroacetic acid. In trichloroacetic acid, if it is used, if any acid contains water, so that water also makes these vesicles insoluble. So that is the reason we find the lipid molecules here and it is not here. So this is the only micromolecule that is found among the macromolecules. Now here you see on the basis of molecular weight, these biomolecules have been we have them. Okay. Now, this was basically the molecules that are present in a cell. Now, in the next class or in the next section of the chapter, we would be dealing with carbohydrate as a whole. See, this carbohydrate is made up of what? Carbohydrate may be monosaccharides, disaccharides, oligosaccharides or polysaccharides. So we will not be dealing with individuals. See, monosaccharides, polysaccharides like that, we won't be dealing with like that. We will be taking up this carbohydrate as an entire unit. Okay.